In today's video, we'll be talking about the mechanism of muscle contraction. Now, when we talk about muscles, one of the important properties of the muscles is to contract and extend, so as to bring about the movement in organisms. Now, when we talk, several scientists were trying to understand the mechanism that how the muscles contract and how they are able to allow the organism to move. So, several theories were put forth, and sliding filament theory, which was proposed by Huxley, is very well accepted, and it explains the process completely. Now, when we talk of sliding filament theory, it is actually a theory that explains the mechanism of muscle contraction, and that is based upon the muscle proteins, that is actin and myosin, and these filaments, how they slide past each other to generate the movement. So in this we are going to understand how this sliding filament theory explain the mechanism of muscle contraction. Now, on receiving the signals from the CNS produces signals, and when the signals are received by the CNS, the motor neurons pass this information or carry this information to the muscle fibers. So when the CNS receives the signal, then those signals are transferred to the motor neurons, and these motor neurons pass this information further to the muscle fibers. Now, the motor neuron, along with the muscle fibers, which is connected to it, that means this motor neurons and the muscle fibers, which are connected, will form the motor unit. They will form the motor unit. And the junction, that means the place where the motor neurons and the sarcolemma, that is the outermost membrane of the muscle fiber, joins, that is known as neuromuscular junction. So the motor neurons, where it joins the sarcolemma of the muscle fibers, that will be known as neuromuscular junction. Neuromuscular junction, or we also call motor end plate. Now, what is happening over here? From the signals received to the CNS pass information to the motor neurons, and the motor neurons pass or are connected with the muscle fibers through the neuromuscular junction. Now, the steps will be followed, which will be taking place as soon as the motor neurons, they reach the neuromuscular junction. A neurotransmitter called acetylcholine. Acetylcholine, which is a neurotransmitter, it is released and it reaches the neuromuscular junction. Now, this acetylcholine binds to the muscle fibers. So, this acetylcholine, it is released when the neural signals are passed. Neural signals passed, it reaches to the acetylcholine. This acetylcholine binds to the muscle fiber, binds to muscle fibers. Now, when it binds to the muscle fibers, this generates action potential. The generating action potential, because before this is in touch, or before the acetylcholine comes in contact with the new muscle fiber, the muscle fiber is in resting stage. So as a result, the action potential is generated. Now, this action potential further, it spreads. It spreads throughout the muscle fiber. So, causes the release of calcium ions. So, it spreads throughout muscle fiber. It spreads through the muscle fiber, and this causes release of calcium ions. And remember, calcium ions are present in the endoplasmic reticulum, that is, we call it as sarcoplasmic cisternae or sarcoplasmic reticulum. And these are released from there into sarcoplasma. That is the cell of that is the cytoplasm of the muscle fiber. Now, this increase of the calcium ions results into the binding of calcium with a subunit of troponin. So what happens? This calcium ions binds to subunit of troponin, and troponin we know is present as a complex having 3 subunits on the actin fiber or actin filament. So what is happening? It binds to the subunit of troponin, which is present on the actin filament, and as a result, this removes the cover or the mask, that cover or mask that prevents the binding of actin to myosin, that prevents the binding of actin to myosin. Now, due to the uncovering of this troponin, that is the subunit of troponin, this causes the binding of, because this is removed, the cover or the mask which is present on the troponin is removed, so the actin is now ready to bind with the myosin. So here the myosin, therefore the myosin binds to actin. And this is, we are talking of the contraction, and it uses the ATP over here, using ATP. Now, as a result, cross bridges are formed. Cross bridges are formed. And due to which, when the cross bridges are formed, this results into the attachment of the actin fiber to the myosin, and they are pulled towards the center of the A band. So if we draw the diagram, that is, we know this is the Z line, this is the Z line, and in between we have the thick band. We mark it as this way. And also we now have This is the Z line, this is the H zone, this is the actin fiber, thin fiber, and this is the myosin. This is actin and this is the myosin. So when the troponin, the mask on the troponin is removed due to the increased concentration of the calcium ions, as a result, the H zone will get reduced and this is the sarcomere. This is the sarcomere. Now, due to the actin fibers being hooked or attached to the myosin, the H zone will get reduced and consequently the two Z lines which are attached to the actin fibers will also come close, causing the contraction. So if we talk about the contraction, that is the process where actin fibers are binding to the myosin fibers, the major steps are the length of I band. I band is what? Actin. It decreases because it will shorten, it will come close and bind to the myosin. Another important highlight is the A band, that is the myosin or the thick band. It remains unchanged. There is no change over here. We are talking of the length. I band length and A band length. There is decrease in the I band or the actin fiber, whereas A band, that is the myosin fiber, there is no change. H zone reduces. And in extreme cases where the extreme contraction is there, then these two will completely overlap each other. They will completely overlap each other, making this H zone completely disappear during extreme contraction. So over here during the extreme contraction, the H zone will disappear, the Z line will move further close to each other. And when we talk of the relaxed state, that means then that time the H zone is having lot of space in between, providing sufficient movement for the actin or myosins to slide over each other. So this sliding filament theory you can understand over here, since the actin fibers are slide passing over the myosin fibers. It's kind of a very smooth, if we talk of this way, suppose this black is the sliding actin fiber and red if we take as the myosin, then the movement of the actin fiber, it is slide passing this and therefore it is referred to as sliding filament theory. Now, we were talking of the contraction. So the highlights of the contraction are that when the signals reach to the CNS, then the CNS through the motor neurons pass the information to the muscle fiber, and as a result the neurotransmitter is being released at that time. This neurotransmitter or acetylcholine causes the change in the action potential or brings about the action potential by release of calcium ions. When these calcium ions are released, because calcium is very very important and stored sufficiently in sarcoplasmic reticulum, so they are released into the sarcoplasma, and as a result, the calcium ions bind to the subunit of troponin, unmasking or uncovering the subunit. And that troponin is now ready to bind with the myosin. So as soon as the cover is removed, this will bind to the myosin, actin filament will bind to the myosin, and as a result the Z line will come close to each other, or the size of the sarcomere will reduce, or reducing of the edge zone will take place, bringing about the contraction. Also, here you have to understand that if the muscle is continuously stimulated for a longer time, then during that time it results into the fatigue of the muscles. So they have to the fatigue can be in the form of cramp or maybe the pain, and it is mainly due to the accumulation of lactic acid, and the lactic acid is formed over there. Lactic acid is the cause of fatigue, muscle fatigue, and it may cause the cramps or all. And this is basically due to anaerobic breakdown of glycogen, because we know glycogen is stored in the form of energy in the animals, and this glycogen needs to be broken down. So when there is less supply of oxygen, that means when the muscles require a lot of oxygen, and if the supply of oxygen is less, then the glycogen will not break completely and will then break into lactic acid, anaerobic breakdown, which cause the muscle fatigue. So over here, lack of O2. Now, when we talk of the relaxation process, during relaxation, the muscle will go back to its original position. Now, the myosin releases ADP and inorganic phosphate. So, during this process, the myosin produces ADP plus inorganic phosphate, and it enters into the relaxed state. This ADP and inorganic will form the ATP. Now, ATP is hydrolyzed by the myosin head, and the cross bridge formation. This is the cross bridge formation over here. So this breaks and resulting into further sliding, and the process continues till the calcium ions, which are present in the sarcoplasma sufficiently, are sent back to the sarcoplasmic reticulum or sarcoplasmic cisternae. So these are the major changes. Cross bridges will break. Cross bridges between actin and myosin will break, and all the calcium will again go back to the sarcoplasmic reticulum, bringing back the original action potential. That means when the muscle is at the resting stage, and the removal of the calcium ions, it also causes the masking of the actin filaments, that is the troponin, and again having that cover, and due to which the Z lines will again go back to their original position, and the muscle will be in the resting stage. So this is the and as again this after the relaxation process, the muscle is again ready to receive the signal to bring about the movement or the muscle contraction. Now, when we talk of the muscle fibers are basically, we classify them as red fibers and white fibers. So when we talk of the muscle fibers, some of the muscle fibers appear red, while some of the muscle fibers appear whitish. Now, the red muscle fibers mainly appear so because they have large amount of oxygen storing pigment. So they have large amount of myoglobin. It is the pigment that stores oxygen, and they also have, since it will have large amount of oxygen over there, they appear red, and this pigment is giving red color to it, and therefore they are known as red fibers. Second important is they have large number of mitochondria. They will be having large number of mitochondria and can use large amount of oxygen, because a lot of oxygen is stored in the myoglobin and mitochondria is ready to utilize that oxygen for various kind of movements. Now, since these muscles use large amount of oxygen to bring about the movement, they are also known as aerobic muscles. So the red muscles are also known as aerobic muscles. When we talk of the whitish type of muscles, they have less amount of myoglobin. So obviously the oxygen content will also be very, very low. And they have few mitochondria. They are just opposite. They will be having few mitochondria, and they are therefore known as anaerobic muscles, because they do not use much of oxygen due to the absence of myoglobin. Myoglobin is present in very, very less amounts, and mitochondria also is very, very less amount. So therefore, they are using the anaerobic method for producing energy. These are the two basic types of muscles, depending upon whether the amount of myoglobin is more or less. So this sliding filament theory is very well accepted theory and is considered suitable for explaining the mechanism of muscle contraction.